All right, folks, we are back and we are on the final stretch. Hopefully, I should maybe not talk so much of the Brian Kohberger Genetic Genealogy Court Challenge. And right now on the stand, we have Daniel Helwig, who was with, at the time, Intermountain Forensics, and he's explaining us all the technological wording process about how it works. Okay, so we talked about he talked a little bit about the difference between STR and SNP technology. As I've talked about before, STR is for CODIS, SNP is for forensic genetic genealogy. Let me introduce myself if I haven't. I'm Christine Burke. I'm a forensic genetic genealogist and retired police detective and sergeant. And I do this stuff all day, every day. So I love talking about these cases and what's actually going on, specifically for my cops and my prosecutors, the challenges that you're going to face, because I don't feel you're prepared for them. And here's a good example, okay? That's why I founded my company, Genetic Genealogy for Law Enforcement, to protect y'all. So, okay. So here's the defense attorney continuing. So she's explaining, I think I understand the repeats as looking for patterns, but the SNP is looking at the individual characteristic. Okay, that's talking about the CODIS. We'll call it the CODIS type of DNA versus the genetic genealogy type of DNA. And he says individual letter nucleotide. So if DNA is made up of billions and billions of letters, STR is looking at repetitive sequences and counting the repeats. And SNPs is looking at specific letters, specific nucleotides on that genome. So she says she wants to talk about the whole process, how you get from down to an SNP from splitting the water balloon open. And that's what he originally described it as, how to get the DNA out of a cell. Okay. So she says, so what do we do after we get that stripped off? Okay. So they take the evidence item, they get the DNA off of it, they clean it up, and then we have to decide, are we going to CODIS, STR type testing? Are we going to FGG type testing, or both? What are we going to do with it? So Mr. Helwig here says, if we're going down the path of forensic SNPs, there's a few ways you can do that. There's one way that is essentially a targeted practice. We're looking at SNPs in question, and we're going to focus on those SNPs and do what is called an amplification PCR. Basically, a DNA photocopier of that sequence, that specific letter, and a little bit around it to specifically target a SNP in question. So they're going to, like he says, the photocopier, okay? Amplification. Think about amplify with sound, louder, right? Right? So they're going to amplify it, okay? Photocopy it. The other technique is something more attuned to whole genome sequencing. Again, there's a variety of different ways you can do this, but the idea here is we're going to sequence the entire genome, the entire length of DNA on this particular extract, and then we're going to pull all the SNPs that we need, the relevant ones that we're looking for. Think about a clothesline sort of thing, okay? Or a string of beads with all the pieces laid out, okay? This can be done, I will refer to that as whole genome sequencing, because they spread out, they sequence the whole thing, they map the whole thing out. And the genome is basically the blueprint for the person. All right, so they print it all out, they lay it all out, they spread it all out, all that laundry, okay, on the clothes rack of the chromosome, right? They put it out there, and then they go and pick the pieces that they need to examine, okay? Like very low level type of explanation, but I'm hoping I can kind of make it a little easier to understand. So, he says we're going to pull out all the SNPs that we need, the relevant ones we're looking for, or, he's going to call that whole genome sequencing. that there's a variety of different ways you can accomplish that. So he says, once you sequence the DNA, it comes off the instrument in question. There's several different instruments you can use, but for the most part, it turns it into raw data. And that data is typically found in a file called a FASTQ file. This raw data is massive. It has an insane amount of information, and it's not refined in a way that we can actually utilize it. So the steps from taking that raw data to actually getting that upload file, that end product for uploading to these databases, is bioinformatics. And it's a software that goes into the massive amounts of data and pulls what they're looking for. She says, is all software the same, or all bioinformatics programs, are they all the same? No. As far as I can tell, each laboratory has their own version of bioinformatics that they use to get from sequence data into usable results. Defense says, if I understand where we are, we popped open the balloon, we put the DNA in a sequencer, we created a raw data file called the FASTQ file, and now we need to do bioinformatics to get a SNP. Right. Okay. What happens from there? So in the bioinformatic pathway, you're doing multiple things. Again, you can think of this as a DNA sequence that the sequencer has given you a truckload of information. They're essentially puzzle pieces. The first thing we're going to do is re-emerging. So we're going to take all the puzzle pieces that are the same, and we're going to use them to paint the best picture of that information. And let's see, I'm trying to give you an analogy here. Forget about the laundry. Let's go to a puzzle. How do we solve a puzzle? He says we're going to take all the puzzle pieces. So think about when we dump a puzzle out onto the table. Now, we know at the end, it's a little different here, but we know at the end what the picture is supposed to look like. But if we didn't, if we just had a bag of puzzle pieces with no picture in our mind, what are we going to do? We're going to take it, we're going to dump it on the table, we're going to start pulling the red pieces to the red pieces, and the blue pieces to the blue pieces, and the white pieces to the white pieces, right? We're going to try to map the colors together and then fit them how they're supposed to go. So it's kind of like this. It dumps all the DNA pieces in there, and then they're going to have to put it together the right way. All right? So it says, paint the best picture of that information. You can think of this as we're sequencing this DNA, the human genome, multiple times, hopefully 10, 15, 30 times. Our samples in forensics are typically difficult, so we don't tend to get that much level of. Essentially, we're trying to sequence this entire genome multiple times to add more context in what we're getting. Okay? Amplify it. In merging, we're taking the DNA sequences that we have multiple copies of, and we're combining them into one, the best fit for that particular fragment of DNA. Okay? They're searching for the best fit, all the red ones. Which one fits, right? Because, round, and what is that, concave, right? The piece has to fit in there. So he says, well, we still have puzzle pieces to put together. The next thing is mapping. The bioinformatic pathway will map all of these puzzle pieces and put them in the genome in the way that they're supposed to be. That takes multiple fragments and lines them up in the right manner. Once it's mapped, we're going to be able to start finding the particular SNPs we're looking for. We have a human genome with a whole bunch of different letters in different positions. We can say, all right, these are the relevant SNPs we're looking for, the genealogy-based informative markers. We want to pull these SNPs out, see what the call is there, and send that to an upload file. Now, remember we talked about medical information versus other information. And our whole body blueprint is on the genome. So she was asking, are you pulling medical information? Are you pulling, what parts are you pulling? So he's explaining, we're only taking the genealogy-based informative markers, okay? And then we're going to put them in the upload file, okay? Or upload file. So what he says is, in some cases, we don't have all the information. We're missing some sequence data. We've maybe gotten 80% of the genome and some of the SNPs are missing. It's like, have you ever printed something on your printer and you're running out of ink in one particular spot? And so, you know, part of the letter is missing, or somehow like on a G, the tail is, right? So you kind of know what's supposed to be there, but it's not showing up, okay? So he says here, there's a process to fill those gaps called imputation. Imputation is in most bioinformatic packages. Essentially, just like our G, we know what the letters are in front of this particular spot we're missing, and we know what the letters are behind what this particular spot is missing. So we can look at all the human genomes and estimate the most likely letter in the position we're looking for. Okay? Once we're through imputation and allele calls, we're going to use this bioinformatic package to pull out SNPs we're looking for and put them into a format that will be usable for upload into these databases. Okay, like I said, when the little piece of that G gets cut off, I was trying to think, you know, I don't know, I don't want to start spelling words, but you, like, depending on the word, like, you know what, you can guess, like, what letter's supposed to be there and what letter's not supposed to be there, because there's not a word for that, right? Like, it has to be one of several possibilities, okay? So, then he says, at IMF, we actually generated two files, one specifically to GEDMatch and one to FamilyTree, because they do a little different computer language, we'll call it, okay. In these files, GEDMatch PRO, we typically got around 550,000 plus SNPs that we were looking for, and the FamilyTreeDNA database upwards to 600 to 650 SNPs. The end product there, though, is a text file or some sort of upload file, which could be in text format or Excel format, but essentially what it is a file with all of those SNPs, the calls, positions, okay, at the SNP locations in a format that allows them to be uploaded into the database. All right, at this point, we hand it off to an investigative genetic genealogist to do the research. So, like I said, you can't just shove a cell or some blood into the machine. It's got to sort it out, clean it out, line it up, fill in any gaps. Then they're going to take only what they need, right? It's like we'll go back to our laundry line. If you need to get dressed to go out, you only need one shirt and one pants or one skirt. You don't need two for that particular moment, right? So they're going to pull the SNPs, the sections only that they need for genetic genealogy. They don't need the rest of the stuff that goes in the closet or it's going to be used tomorrow or whatever for something else. They're only going to take the pieces that they need for genetic genealogy. So she's asking him, the upload databases you mentioned too, GEDMatch PRO and FamilyTreeDNA. That's correct. Why did you mention those? Those are the two databases that allow law enforcement searches. They have specific terms of service that give a portion of that database, those that have consented to do so, to law enforcement to search in for human remains in some cases and in criminal cases. And I want to point out here, he says GEDMatch is one database. GEDMatch PRO is one database. He's saying a portion of that database. Now, we heard earlier that it's 2. I could be wrong. But I believe that they just developed a different GUI, what they call graphical user interface, to access only those that opted into law enforcement searches in GEDMatch PRO. I don't believe that they replicated the data. I could be wrong. So it may only be one database, but I'm not sure. Okay. So when you go into GEDMatch PRO, you're supposed to be only seeing the people that opted in. Okay. You may not know that, like in Ancestry, just the regular databases, forget about law enforcement, I can upload my DNA as a person, and it could be resident in the system, but I could choose not to match with anybody. It could still be in there. So in the GEDMatch database and the FamilyTree database, they're going to be matched, is one thing. And then only some of those people are going to say cops can look at my matches. Okay? So, he talks about because it's for human remains in criminal cases. So the defense asked, so in this lab process, to get the SMP to the upload file, it sounds like there's a lot of steps along the way. He says it's pretty complicated. She says, is it important to keep lab notes or bench notes or have procedures or protocols? Where have we heard that before? He says, yes, and I think that's pretty standard practice amongst forensic DNA testing laboratories in general. You're going to adhere to a quality management system. You're going to have procedures, protocols, and policies that tell you what to do, tell you how to do it, tell you what samples to use, what personnel can do the analysis and technical review, and so on and so forth. Who has access? It's a whole quality structure that gives support and credence to reliable results, and that would include protocols, SOPs, case documentation, lab notes about what you did, how you did it, chain of custody documents, electronic records as needed, and hopefully at the end of the report, it includes conclusions and information about what was done. Remember, the defense asked about the Othram report that didn't say nothing. Okay? It talked about those brothers. All right? But they've testified, other people have testified. It didn't say how they came to the brothers. Okay? So she's asking Mr. Hellwig, why do you want to do all that? He says, first of all, within the forensic DNA community, and that's any kind, it's part of our accreditation process. Here we have accreditation and some of the standards that we adhere to as a community. Second, it's just best practice. The holding of the records, using them, creating them, and retaining them. You never know if you're going to need to refer back to them in your own notes. Or we mandate a technical review of all this data so a second quality analyst will look over all of your laboratory notes, all the work you did, and your reports, and make sure they agree with it and would come to the same conclusions for discovery requests. Now remember, when I talk to you guys about, as a forensic genetic genealogist, within the same company, within the same police department, within a prosecution versus defense, I should be able to follow your methods and come to the same conclusion. Because the science is the science, okay? So that's what he's talking about here. Now, there's been problems in labs, I can't remember the lady's name specifically, in Colorado, and there were problems. Nobody was what they sometimes call dual control. Nobody was QCing, quality controlling, and reviewing her stuff, and they had to throw out tons of cases. It's Colorado, okay? So you guys can look for that. I don't think I have a video on that on my YouTube channel. I do have it in my genetic genealogy training, okay? Because it's problematic, okay? So he says at his company, they have typical documentation that they would send to the clients. which would include a report, all the case notes, and lab documentation associated with that. Outside of that, he often gets requests for more information. What are the validations? What's the quality manual? Competency tests for our personnel, their training and education, a whole list of support documents that are within our quality management system that are also important in accreditation of our laboratory. So again, remember we talked about, if you haven't watched the other videos, please go back and watch those videos. Accreditation. It's not just a lab. So do labs need to be accredited? Do they not? Did you know police departments, most police departments are not accredited. It costs a lot of money. It's a lot of hassle. Speaking as a cop and an agency. Costs money, a lot of hassle. Is it worth it? So most agencies are not accredited, but they have SOPs and procedures and best practices in place. But they can go get accredited, and then they have additional rules to maintain that accreditation. So, watch the other video. Okay. So, before you became a 501c3, was your laboratory accredited? He says, even prior to becoming a 501c3, since the inception of the company, even before we no longer did lab operations, we got accredited by ANAB ISO 17025. They've got these types of accreditations. He says that was on our accreditation scope, including traditional forensics, STRs, YSTRs, which is CODIS. And then he said they included eventually forensic SNPs over to genetic genealogy, massively parallel sequencing that encompasses accreditation for this whole genome sequencing in support of FGG. So she says, is there a set of guidelines or rules that apply to work with DNA to split that DNA water balloon open? He says, yes. And I think you've heard testimony to that. There's accreditation standards. ISO 17025 is the big one, as well as the Federal Bureau of Investigation Quality Assurance Standards. For forensic DNA testing laboratories, those are common. I would say more so almost required within the forensic DNA community, and that's just not FGG. And those are standards that dictate our quality management system as a whole, what we do, how we do it, how we support it, how we document it, and how we report it. She says, is that important so there's public trust in what you do? He says, more than public trust, reliability and reproducibility of results, transparency, and just ensuring a full quality behind your conclusion and reports. Let's just talk about a cooking recipe. If all the parts of the cooking recipe, your neighbor is having this fantastic flan, for example, and I don't even know how to make that, but let's just say she forgets to tell you that she's using parchment paper or something, or a certain type of pan, and you try to make it, and she leaves that out on the recipe, and she tries to make it, she's not going to get the same results, and hers is just going to be crappy. And she's like, I'm doing everything that the recipe said, but I'm not getting the same result. Okay, so it's just making sure all the steps are there, right? So now she's asking him about the SNP profiles for GEDMatch PRO and FamilyTree. That FamilyTree or GEDMatch 550,000 or 650,000 seem to be the maximums. So she says, if you had a case where you had produced a file to upload, an upload file, and it had, let's say, 600,000 in it, could you take that text file and turn it into twice that big? And he says, I'm not sure how we would do that. No. He continues. An important note here, that was just IMS policy to get to the 500 or the 650, 500, 600-ish. He says we generated upload files specific to each one of the databases, so meeting their recommendations. He says, and that's the number we had within our bioinformatic package. That's not necessarily what would be true of others. Okay? So remember when we talked about accreditation for the police department, let's use, you have to meet the accreditation, somebody else telling you what their standards are. Okay, so in this case, that would be GEDMatch PRO FTDNA, telling their numbers. But so let's just say they took a minimum of 100,000. Well, in his IMF, in his lab, he also has his own policies. And he could say, I know they only wanted that 100,000, but we're going to every time hit 400,000. Okay? Like, hopefully that makes sense. So he says that the 500 and the 600 were just IMF's policies. They generated files specific to each one of the databases, and that's the number that they programmed within the bioinformatic package. That's not necessarily true of others. That being said, if theoretically we were able to deliver our upload file to our client and say it had 500,000 SNPs on it, I don't know of a mechanism to add SNPs in that manner without going back to our raw data. They're either there or they're not there, is I guess what he's saying. So she says, in this particular case, I know you haven't gotten involved in all the ins and outs, but we did provide to you what we received in regular discovery, the Othram file and the FBI file, upload files. Yes. I did receive 2 upload files. One was a text file that came from Othram Labs, and one was an Excel file that came from the FBI. Is that also an upload file? Yeah, both of them are upload files. They contain exactly that, a listing of SNPs, the call, and their location in a format that would allow them to be uploaded to databases. What was the difference in size? The FBI upload file had significantly more SNPs called than the Othram file. I wasn't able to go deep into it. I don't know exactly how many more, but there was more SNPs called within the upload file for the FBI than there was with the Othram file. She says, and since the Othram file is smaller and it's a text file, can you tell how the FBI got so much more data, so many more SNPs? Because remember, the computer's reading the thing and then pulling them. He says, no, that's kind of the important part here. There's no documentation to say how this happened. And that's really the important part, right? I have no reason to believe that Othram Laboratories did anything wrong to obtain their profile. I just don't have the documentation to show how that came to be. Same with the FBI file. There's no documentation or notes or bench report to say this is what I had, this is what I did, and this is what I now have. So I'm unsure of how that came to be. So she says, thank you, that's all I have. So did the FBI output a brand new file? Did they take the DNA and output their own file? Like, did they do their own laundry? Did they wash the laundry again? Right? Othram washed the laundry and hung it up. And then they went and picked which shirts they wanted for their trip. Good analogy, maybe. And then the FBI did their laundry. And they said, well, you know what? We're just going to take 15 things because we don't know what we're going to wear. We're going to take more. So, started from the same data, but the laundry was done differently. We just don't know. That's what he's saying. We don't know. We don't know what the hell happened. Okay. Now the prosecutor's going to cross-examine him. So he talks about his accreditation testimony, or his accreditation, that the lab was accredited since inception in 2019. He clarifies traditional forensics, STR, and other things, not FGG. He says, We began the accreditation in 2019, and I believe the first accreditation scope was in 2020. For forensic SNPs, our accreditation was January 20th of 2023. So he says you weren't accredited back in 2019 for forensic SNPs, correct? Correct. We weren't doing SNPs at the time. But you were working on forensic SNP cases prior to your accreditation, correct? He says, no, we didn't work on those until our accreditation. January of 2023. Interesting. Side note, okay. Side note, okay. Okay, do you recall describing accreditation process as a chicken and egg problem? I don't. You testified you work at Intermountain Forensics. That's correct. Do you post on their website? Yes, sometimes. Do you post in your name? Certainly. You don't recall posting in June of 2020 about accreditation being a chicken and egg problem. I don't recall. I'm certain that the statement is on there. I would need to recollect, certainly. I'm happy to show it to you. So he shows it to the defense attorney, shows it to him. He said, okay, yeah, I remember it. So he says, yeah, it was several years ago. And the state is asking, in that same post, you talked about how you appreciated the trust of those that were using you. Ding, ding, ding. Before you were accredited. Side note. Correct, yes. Do you feel like your work before accreditation was less, a lower quality than after accreditation? No, absolutely not. Important. Prosecutor, I also want to talk to you about, you mentioned that Intermountain Forensics does, or at least used to do some cases like this, right? Yes. Assisting law enforcement, yes. Roughly how many cases did you assist in? That's a good question. We have a running total in there, but I'm guessing in the hundreds. Okay, and what was your role personally in assisting law enforcement in those cases? I had several. I did consultations up front. I was qualified as an analyst for forensic SNPs. I did some technical review, and in some cases, I distributed files to our clients. And when you say in the hundreds, you meant you went through that whole process you talked about with Miss Taylor in hundreds of cases. Yes. Developing a SNP profile. I believe so, yes. Interesting. Okay, and in all of those cases, were you successful in developing a SNP profile? No. In roughly how many cases were you successful in developing a SNP program? Is this program? I think they mean profile. We were about, at the end of laboratory operations, we were about 80%, somewhere in that range. So 80% of hundreds. I believe so. In the cases you successfully developed a SNP profile, did you learn medical information about the individuals whose DNA you were looking at? No. Did anyone at Intermountain Forensics learn medical information about those people? No. When you develop, I believe you called it an upload profile, is that correct? Yes. What does it look like if you just open it on a computer? It's a text file, a very, very big. Again, it comes in different formats. But essentially it's a text file. It's a big text file that has hundreds of thousands, in our case of SNPs, that have a position, a name, what chromosome they're found on, and the actual letters of what we're looking for. So, if you provided that upload file to someone that didn't have any training or experience in genetics, would they be able to read it? No, not in so many words. There's some headers on there, I guess, that you could, that would be narrative. But the molecular biology would be pretty complicated. Let me ask it this way, I guess. If you were to hand that upload file to your average police officer and ask them whether or not the person whose DNA you are looking at would have cancer, would that officer be able to do that? No. What additional steps would you need to take to learn medical information from a SNP program above and beyond what you were doing at Intermountain Forensics? He says, well, SNPs are very are informative to a variety of different things. So what we're looking at is genealogy informative markers or ancestry link informative markers. To find health information or phenotypical, like hair color, eye color, you need to look at different SNPs. So remember when I said going off the laundry line and picking the outfit that you need for that day, instead of packing up all the clothes, that's how it is. Okay? So when the defense in this case was arguing about the medical information, they didn't pull those SNPs. Okay? That's what he's explaining here. So if I'm understanding correctly, at IMF you developed SNP profiles, but they were specific for ancestry. Genealogy and ancestry, yes. Why didn't you develop SNPs related to medical information? That's not part of our scope. We are a forensic DNA testing laboratory. We're generating FGG profiles for the support and upload into these genealogy databases. It wouldn't be relevant, probative, or appropriate. Did you, when you helped these law enforcement agencies, did you ever follow those cases to see if there were successes at the end? Sometimes. We delivered files to our partners. Interesting. In some cases, we had forensic investigative genetic genealogy team that worked the cases on the investigative side. But in some cases, our clients would come back to us and let us know they've gotten a result or they've had a case resolution. And you participate in the, I don't know exactly what you called it, the FGG community. Would you consider yourself a member of the FIG community? Yes, I guess yes. There's a variety of different groups, I guess, message boards and information pathways. I try to be a part of that where I can. And as a member of the FGG community, did you hear about other people's success stories using FGG? Absolutely. Have you ever heard of an FGG case that was successfully solved using medical information? No, never. So then he checks any questions. He doesn't have any more. That was the prosecutor. So here comes the defense on recross or redirect. So Ms. Taylor asks, full genome sequencing is the whole genome sequence, the whole genome, everything. Yes. It's the attempt to sequence the entire thing if possible. She says, now I heard you talking to the prosecutor about what you didn't look with the medical information, but I thought I heard you talk about when you got down to the SNPs, those SNPs were not called from the whole genome. Did I hear that right? He says, I would have to rephrase that a little bit. When we're dealing with whole genome sequencing, it's a whole blueprint, we're attempting to sequence the entire genome, and it turns into that raw file, that FASTQ file I mentioned before. The next step is the bioinformatics. That only targets specific SNPs to what they want to solve the question for. In our case, these are genetic genealogy or ancestry informative markers. They would be specific to those SNPs, but the raw data, the sequence data, still has the entire list of SNPs. We're just not pulling that piece of information out. She says, so if you handed me a case with your notes, protocols, and procedures, I could look at what you did at whole genome sequencing and know you were targeting only genealogy SNPs. Would that be fair to say? With the right knowledge base, yes. The list of SNPs are included, where they are, and the name on the upload file are all there. You can do the research to look at that and say, okay, these are genealogy or ancestry informative markers. So she says, if I had a file with notes that talked about how bioinformatics was used, what to produce, what kind of SNPs to produce, and handed it to somebody like you, could you tell me these are the ones that go to ancestry issues, that go to genealogy issues, if I had the notes from somebody else? So I want to say different parts on your chromosome do different things. Okay, that's how they locate different things. That's why they're only looking over here for some things over here. You know, genealogy's over here, cancer's over here. Like, you know, a house versus a business. all right? You know, your checkbook is probably in the office. It's not in the refrigerator, okay? So he says, you know, looking at those notes, he says the SNPs that we're using are derived from databases. So it's GEDMatch PRO, FamilyTree, Ancestry.com. Those are the ones who have decided which SNPs to use. Where on the clothesline, where on the genome. And we just use them as a tool. It says, look here, look here, look here. So what is the SNPs that best upload into the FamilyTreeDNA law enforcement database or GEDMatch PRO? And so when we are bioinformatically pulling out the SNPs, we're doing so under the direction of what they want from us. Go here, go here, go here. It's kind of like a DoorDash. I feel like, you know, a sandwich today. Go over here. They're not going to the doctor. They're going to the sandwich place. He says, could I determine if there's medical information within those SNPs? With medical information, yes, but it would be a lot of research. She says, I just have a couple questions for you about what the prosecutor talked about besides that. He asked you about the chicken and egg problem in different cases you've done and the hundreds of cases you've done before. You told us it's important to have protocols and procedures and keep your information, keep your notes. Did you do that in those cases? Oh, yes, yes. Within the quality management system as a whole and adhering to our procedures and protocols and the accreditation standard that we derive to. It's all that. It's case documentation, laboratory report, and the foundation behind it. Validation summaries, competency testing for personnel, their educational requirements, all that has done in those cases and adhered to in our own internal policies. Basically, any test, anything that they do, all should follow the same thing. Whether they got a good result or not. Any work should follow the work rules, okay? So she says, okay, that's it. You can step down. Do they have any other witnesses? She says she has one witness, and they're going to go to lunch. So we are going to end this one here. All right, we are almost there, folks. We're almost there. Next, we're going to talk to Leah Larkin.